Yes, welcome back for once for our 12th lesson in cell physiology. And the objective of our study today is to state the differences between osmosis and diffusion, look at the two processes and compare, and then contrast, and also to define the term called osmotic pressure and osmotic potential. Now, what are the differences between these two processes, that is, osmosis and diffusion? Now, we are going to concentrate on this table. We're going to concentrate on this table and see the differences. The first differences between the two processes is that diffusion involves the movement of gas or solid particles. So when you open a perfume, and then the perfume molecules will fill the room, the smell, it is the molecules of that perfume that are moving. We have gases which have got different smells, and normally they spread all over from where their concentration is high to where their concentration is low. So diffusion involves the movement of gas particles. Now, another thing about diffusion is that we have seen diffusion taking place in solution form, meaning the particles of a solid dissolve in a liquid normally diffuse from where their number is high, from where their concentration is high to where their concentration is low. So we say diffusion involves the movement of gas or solid particles. But osmosis only involves the movement of solvent molecules. That's why we define osmosis as the movement of water molecules from a region of high water potential to a region of low water potential through a semi-permeable membrane. Or you can say that osmosis is the movement of solvent molecules, meaning liquids, from a region of low concentration to a region of high concentration, or from a lowly concentrated solution to a highly concentrated solution through a semi-permeable membrane. That's one difference. The second difference is where they occur. So diffusion may occur in air, particles may diffuse in air, that is gases, or in liquids or through membranes. So when diffusion occurs through liquids, we say it takes place in solutions. Or it can take place through membranes, through a cell membrane, plasma membrane, mitochondrial membrane, subvacuole membrane, or tonoplast. So that is diffusion. But what about osmosis? Osmosis occurs only through semi-permeable membrane. I want you to note, for diffusion, it is any membrane, whether it is semi-permeable or whether it is fully or freely permeable. Freely permeable means a membrane which can allow both substances with small particle size and those with large particle sizes to pass, while semi or semi-permeable, simply it can only allow those substances with a very small particle size to pass across it. Then the third difference is that diffusion is not affected by something called pH changes. So pH changes does not affect the process of diffusion, but the pH affects the process of osmosis. So osmosis is affected by pH because osmosis can only occur across membranes, semi-permeable membrane. Remember we say the semi-permeable membrane, which is the cell membrane or any other membrane within the body or within the cell, like the membranes that cover the cell organelles. But remember, the cell membrane is made up of proteins. Proteins are sensitive to changes in pH, and therefore it means that anything that affects the cell membrane will affect the process of osmosis. We move on to step 4, where we are going to discuss something called osmotic pressure. Now, when we talk of osmotic pressure, it is the pressure that must be applied against a semi-permeable membrane to stop water molecules from the distilled water entering the concentrated solution. I want to demonstrate that the whole concept, that the pressure that must be applied to stop, or the pressure that must be applied against a semi-permeable membrane to stop water molecules from the distilled water getting into a concentrated solution. Or you can say, it is the pressure that must be applied against the cell membrane to stop osmosis from taking place. What is this? Now, let us take a beaker and divide this beaker using a semi-permeable membrane. Using a semi-permeable membrane. You can see it is semi-permeable. Then on one side, we place pure water. This is solution A, and this is B. So this B is pure water. This is pure water. Let's call it distilled water. On this side, we have a salt solution, sugar. So let's say we have sugar molecules. So we have sugar molecule. Sugar molecule. This is water molecule. Now, so we know that when two solutions are separated by a semi-permeable membrane, then water molecules will move from where we have more water molecules to where we have less water molecules. So water molecules will move by osmosis from a solution of low concentration. So here we can say there is low concentration. But here we have, this solution is higher concentration. Or we can say we have high water potential because we have more water molecule this side. And where we have less water, we say low water potential. This one is semi-permeable membrane. So we have agreed earlier that whenever two solutions of different concentrations are separated by a semi-permeable membrane, then water molecules will move from a solution which has lower concentration to one which has got a higher concentration. Meaning, if it is of low concentration, it means it has more solvent, more water molecules, to where we have less water molecules. Because on this side you can see the water is mixed up with sugar. So we are saying that the pressure applied, when we talk of osmotic pressure, like what we are just discussing now, the osmotic pressure. So when we talk of osmotic pressure, we are talking of the pressure that must be applied against a semi-permeable membrane to stop water molecules from the distilled water entering the concentrated solution. We can stop water from moving into the concentrated solution. How do we stop? By applying pressure. So that pressure which is applied against the semi-permeable membrane to stop water molecules from moving from the distilled water into the concentrated sugar solution is what we call osmotic pressure. The pressure which is equivalent to the one that must be applied to stop osmosis from taking place. So that pressure. Now, the idea is one. When you separate these two solutions, the sugar solution will develop a suction pressure, pulling, sucking. This sugar solution, which is more concentrated, will start to suck water molecules from the pure water. So if there is a suction pressure, we can stop this movement of water by applying osmotic pressure. So that one brings us to the first segment, and in the second segment we are still going to continue with the same concept. Let us meet in the second segment.